Welcome to the Deep Dive. We're the place you come to get the real story behind complex science. Today, we're tackling something huge. Aging itself, not just the years you've lived.
Right, not your chronological age. We're talking about biological age, how old your body really is internally.
And the difference can be, well, massive between people born in the same year. We used to just guess why, but now...
Now we have tools, specifically these things called epigenetic clocks. They're kind of revolutionary for measuring how fast, biologically speaking, you're moving down the road.
So that's our mission for this deep dive. We've got the latest research on these clocks, think GrimAge, Dundon Pace, and we're looking at two really interesting human studies through that lens.
Yeah, one looked at slowing aging down with diet changes, calorie restrictions specifically.
And the other, even bolder, trying to actually reverse age using a combination of drugs.
So the big questions are, can we measure our aging speed accurately? And, the really exciting part, can we actually change it? Maybe even turn back the clock.
Let's get into it.
Okay, so before we talk clocks, maybe just a quick refresher on biological aging itself. What is it, really?
Good idea. It's not just wrinkles, right?
Definitely not. At the molecular level, it's damage piling up, your DNA gets hits, proteins misfold, those protective caps on chromosomes, telomeres, they shorten.
And you get those zombie cells, senescence.
Exactly, senescent cells. They stop dividing, but hang around, causing inflammation. So to measure all this, science now looks at something called DNA methylation marks.
Okay, DNA methylation. Break that down for us. What are these marks?
Think of them like little sticky notes or tags on your DNA. They don't change the actual genes, your underlying code, but they tell your cells which genes to read and which ones to ignore or how loudly to read them.
Like volume knobs for your genes?
That's a great way to put it. And critically, the pattern of these tags changes as we age, predictably. We can read those patterns. That's the basis for these epigenetic biomarkers.
Which brings us to the clocks themselves. You mentioned there are different kinds. The car analogy might help here.
Yeah, let's use that. The first main type, including clocks like PhenoAge and the really important one, GrimAge, they're like the odometer in your car.
Okay, tells you the total mileage, how far you've gone.
Exactly. They estimate your accumulated biological age, how much aging damage you've sort of racked up over your lifetime.
But then there's the newer type, the speedometer.
Right. That's where something like Dundon Pace comes in. It's not focused on the total mileage, but on your current speed. How fast are you aging right now?
That seems incredibly useful. Knowing the speed lets you know if you need to slow down.
Precisely. It measures the rate of biological wear and tear. Are you cruising along gently, or is your foot slammed on the accelerator, biologically speaking?
Let's zoom in on GrimAge for a second, though, because you said it's highly predictive. What makes it so powerful? It's not just counting age tags, is it?
No, it's much smarter than that.
Yeah.
GrimAge is a composite. It takes your chronological age, your sex, and then adds in DNA methylation estimates for 7 specific proteins found in your blood.
Like which ones?
Key ones like PI1, GDF15, things linked to inflammation, clotting, cellular stress. Plus, it even includes a DNAm-based estimate of how much someone has smoked over their life.
Wow, so it's pulling in direct measures of health risks?
Exactly. High GrimAge score. That's genuinely grim news, as the name suggests. It's incredibly strongly linked to lifespan, healthspan. I mean, statistics are wild. It predicts time to death with astronomical certainty. We're talking p-values like 2.0 times 10 to the minus 75. That's not just noise.
So PII1, for example, why is that protein marker so critical that it's built into this super predictor clock?
Well, PAI1, that's plasminogen activator inhibitor 1, it's deeply involved in blood clotting, tissue scarring or fibrosis, and chronic inflammation. So by tracking its epigenetic signal, GrimAge is directly linking your aging rate to your risk for things like heart disease, metabolic problems.
It's connecting the dots between the molecular level and real-world health outcomes.
Perfectly put. And GDF15 is similar. It's a stress signal that goes up when cells are damaged.
It's amazing. And now researchers are even pushing beyond just prediction, right? Trying to find the causes. Things like causage, damage.
That's the frontier. Moving from clocks that track correlations to clocks designed to pinpoint the epigenetic changes that might actually drive the aging process. That's, a huge step for finding ways to intervene effectively.
Okay, so if these clocks are so good, especially GrimAge showing us the risks, it really underscores why finding interventions is key. Let's talk about the first big human trial. Can we actually slow down that aging speedometer? Our sources cover the calorie of phase 2 trial.
Right, calorie. This is a big deal. The first time long-term calorie restriction, or CR, was properly tested in healthy, non-obese people. About 220 folks, followed for 2 years.
And the headline result, did it work like it does in, worms and mice?
It did. They confirmed it in humans.
Mm.
CR successfully slowed the pace of aging.
But there was a huge catch. Methodologically speaking, it almost looked like it didn't work initially.
That's a critical point.
Hmm.
When they looked using the older odometer clocks, PhenoAge, even GrimAge, they barely saw any change. Nothing significant.
So if they'd only used those clocks.
They might have concluded CR doesn't work in humans, or the effect is tiny. Years of research potentially down the drain. It was only when they applied the speedometer clock, DNAm pace, that the effect became clear.
It really highlights how crucial using the right tool is, measuring speed versus distance.
Absolutely. Makes you wonder how many past studies on diet or exercise might have missed real biological effects because they were using odometer clocks, not speedometers.
So what did DNAm pace show? How much did CR slow things down?
The data showed about a 2 to 3% slowing in the pace of aging in the CR group compared to the control group.
Okay, 2 to 3%. That might not sound like a lot initially.
But translate that into risk reduction.
Hmm.
Based on other population studies, that degree of slowing is estimated to lower your overall mortality risk by something like 10 to 15%.
Wow. That's actually significant. What's that comparable to?
It's in the same ballpark as the mortality benefit you get from successfully quitting smoking.
Okay, that puts it in perspective. So it proves slowing human aging via lifestyle is achievable, even if, as the sources say, cutting calories significantly is probably not for everyone.
Right. It's tough to maintain long term. But calorie gave us that vital human proof of concept. It showed us what slowing aging looks like at the molecular level using these advanced clocks. Now we can use that benchmark to test other things, maybe intermittent fasting, specific supplements, things that might be easier to stick with.
Okay, so slowing is possible. What about the Holy Grail, reversing age? Let's get into intervention 2, the TRIIM trial. Sounds intense. Thymus regeneration, immunorestoration, and insulin mitigation.
Yeah, TRIIM was incredibly ambitious. A pilot study, mind you, but with a huge goal.
Yeah.
Trying to regenerate the thymus gland in men aged 51 to 65.
The thymus, that's the gland that trains our T cells, part of the immune system, and it shrinks drastically as we age.
Exactly. It involutes, basically shuts down over time. And that decline is a major driver of what we call immunosenescence, the aging of the immune system, making us more vulnerable to infections and cancer.
So what was the TRIIM protocol? How did they try to regrow it?
It was a combo therapy. They used recombinant human growth hormone, RHGH, which was thought might stimulate the thymus, but GH has potential side effects.
Right, I remember reading it can mess with insulin sensitivity, potentially increase diabetes risk.
Precisely. It has a known diabetogenic effect. So very carefully, the researchers added 2 other drugs, DHEA and metformin.
Ah, metformin, the diabetes drug, and DHEA.
DHEA is a steroid hormone that declines with age. Both were added specifically to counteract that insulin glucose risk from the growth hormone. It shows they were thinking carefully about safety and managing the intervention.
Okay, so did it work? Did they actually see the thymus, plump back up?
They looked using MRI scans, and yes, they saw a significant increase in what's called the thymic fat-free fraction, or TFFF, meaning less fat and more functional tissue in the thymus area. It strongly suggested they were achieving actual tissue regeneration, restoring functional thymic mass, like waking up that dormant immune factory.
Incredible. Did they see other positive health signs beyond just the thymus itself?
They did. Several markers improved systemically. For instance, the prostate cancer risk indexed the ratio of total PSA to the percentage of free PSA. It actually improved significantly. That benefit even stuck around after the trial.
Interesting. What else?
They saw C-reactive protein, CRP, go down. That's a key marker for inflammation throughout the body. Lower CRP is good news.
And kidney function, too. You mentioned EGFR.
Yes, estimated glomerular filtration rate, a measure of kidney function, also improved.
Why focus on kidneys in an anti-aging context?
Because kidneys take a real beating from aging, inflammation, metabolic stress.
Yeah.
Seeing EGFR improve suggests the treatment wasn't just hitting the immune system in isolation. It was having broader, potentially rejuvenating effects on organ function and reducing systemic damage. It's a strong sign of a whole body benefit.
All right, this is leading up to the big one. The thymus looked better, other markers improved. What did the epigenetic clocks say? Did they reverse age?
This is where it gets really wild. Yes.
Yeah.
Across the 4 different epigenetic clocks they used, the treatment resulted in a significant reversal of epigenetic age.
How much reversal?
On average, after the 12 months of treatment, the participants' epigenetic age was about 2.5 years younger than when they started.
2.5 years younger biologically in just 1 year. That's profound. But we need the caveat, right? Small study.
Absolute critical caveat.
Right.
This was a pilot study with only, I think, 9 men completing it fully. Tiny sample size, so we have to be cautious about extrapolating too much.
Okay, duly noted. But even with that caution, the direction of the effect reversal is still a massive finding. Did they see anything about the rate of reversal?
They did, and this was also fascinating, though again, small numbers. It looked like the reversal accelerated over time. The average rate was about minus 1.5 years per year for the first 9 months.
So aging backwards at 1.5 times normal speed?
But then, in the last 3 months of the trial, that rate jumped dramatically to almost minus 6.5 years per year.
Whoa. 6.5 years of age reversal per year.
Again, tiny sample, needs replication. But it hints that maybe once the body's regenerative processes kick in past a certain point, the benefits compound or speed up. Very speculative, but intriguing.
And did the effect last? What happened after they stopped the treatment?
This is perhaps the most stunning part. They checked again 6 months after the men stopped taking the drugs. And the age reversal measured by GrimAge, that highly predictive clock we talked about, it held.
It persisted. The 2-year gain was still there half a year later, with no more drugs.
That's what the data showed. The epigenetic age reduction seemed to stick, at least for that follow-up period.
Okay, let's connect this back to the biology. What was happening with the immune cells to explain this apparent age reversal and thymus effect?
The cellular data backed it up nicely. It really pointed towards reversing key aspects of immunosenescence. Two main things jumped out. First, the lymphocyte-to-monocyte ratio, or LMR.
LMR higher is better,
Yeah.
right? Associated with better outcomes in lots of diseases.
Exactly. A higher ratio of lymphocytes, like T cells, to monocytes, involved in inflammation, is generally a good sign. The TRIIM treatment caused a significant and, importantly, persistent increase in LMR.
Okay, that fits. What was the other key immune finding? Something about NAD plus D. That molecule gets a lot of buzz in anti-aging.
Right. It ties into the monocytes. See, certain monocytes, especially ones that increase with age, express a lot of an enzyme called CD38.
And CD38 is bad for NAD plus.
It's the main consumer, the main degrader of NAD plus outside of our cells' powerhouses, the mitochondria. Think of NAD plus as essential fuel for cellular repair, energy, everything. CD38 chews it up.
So more aging monocytes means more CD38 means less precious NAD plus fuel.
You got it. The TRIIM treatment led to a decrease in these CD38 positive monocytes. The implication is that this helped preserve the body's NAD plus levels and cut down on that age-related inflammation driven by monocytes.
So it wasn't just making new T cells via the thymus, it was also cleaning up the neighborhood, protecting the fuel supply.
That's a great analogy. And yes, they also saw increases in naive T cells, the fresh recruits coming out of the regenerated thymus, both CD4 and CD8 types. It all fit the picture of restoring youthful immune function.
This is amazing looking at these big interventions, but the sources, especially around GrimAge, also offer some really practical, almost granular insights from those DNA methylation markers themselves, right? What else are they telling us about what drives aging?
Definitely. We can circle back to DNAm PI1, that marker built into GrimAge. The data shows it's an incredibly strong age-adjusted biomarker, particularly for metabolic health issues.
Okay, so PII1 again. What specific lifestyle factors or health problems show the strongest link to a high DNAm PII1 level?
The correlations are striking. High DNAm PI1 goes hand in hand with high triglycerides, high insulin, high glucose levels, classic signs of metabolic syndrome.
The kind of stuff linked to poor diet and inactivity.
Precisely. And even more directly, it showed strong associations with actual physical measures from CT scans.
Yeah.
Specifically, with measures of fatty liver disease and the amount of visceral adipose tissue, or VAT.
VAT, that's the dangerous belly fat deep inside, around the organs.
That's the one. So a high DNAm PAI1 score isn't just some abstract aging number. It's reflecting, at the epigenetic level, the biological consequence of carrying that harmful internal fat. It links the molecular clock directly to things like diet and exercise habits.
That really brings it home. And what about the smoking marker in GrimAge? You said it was powerful.
Yeah, the DNAm pack years predictor. Get this. It turned out to be a stronger predictor of lifespan than people's own self-reported smoking history.
The clock knows better than the person how much they smoked. or maybe how it affected them.
It suggests the biological damage or maybe an individual's inherent susceptibility to that damage from smoking or other pollutants leaves a deeper, more accurate epigenetic scar than just asking someone how many packs they smoked.
That's incredible.
And even weirder, that DNAm smoking marker predicted mortality risk even in people who reported never smoking.
Seriously, how could that be? Secondhand smoke? Pollution? Or just some underlying biology?
Could be any of those. Maybe capturing hidden environmental exposures, or perhaps it reflects an innate biological sensitivity that makes someone more vulnerable to lung issues or other aging damage, even without smoking. We don't fully know, but it's fascinating.
And finally, linking the overall aging rate, age-XL-GRIM, to broader lifestyle factors.
The connections were exactly what you'd expect, but now with epigenetic proof. Faster aging, higher age-XL-GRIM, was linked to diets higher in fat and lower levels of carotenoids in the blood.
Carotenoids. Those are the pigments in colorful fruits and vegetables, so a marker for veggie intake.
Exactly. Low carotenoids basically means not eating enough vegetables. On the flip side, things associated with slower aging, a lower age-XL-GRIM score, higher levels of education, and, no surprise, regular physical exercise.
It paints such a clear picture. All the healthy habits we kind of intuitively know are good for us: eating well, moving your body. They are demonstrably slowing down our fundamental aging clock at the molecular level, measured by these incredibly precise tools.
It provides that rock-solid biological why.
Okay, so let's wrap this deep dive up. We've explored these incredible epigenetic clocks and seen two potential paths forward based on the human trials.
Right. Path 1, shown by calorie, is that we can definitely slow the pace of aging. Sustained lifestyle changes, like calorie restriction, make a measurable difference, and we can track that progress using speedometer clocks like DNAm pace.
Slowing the speedometer. Makes sense.
Path 2, hinted at by the ambitious TRIIM trial, is potentially more radical, actually reversing biological age using targeted interventions like that drug combination. And we can measure that reversal, that odometer rollback, using clocks like GrimAge.
So slowing versus reversing. Two different strategies measured by two different types of clocks.
Pretty much. And TRIIM gave us that tantalizing glimpse of reversal, with the effect even persisting after treatment stopped.
Which brings us to the final thought, the big question left hanging by TRIIM, especially given its small size. That persistence. The 2-year age reduction measured by GrimAge sticking around for 6 months off treatment. Was that gain truly locked in?
Exactly. Was that 12-month intervention enough to permanently reset their biological age trajectory by 2 years? Or, to maintain that benefit, maybe even continue the reversal, would they need to repeat the treatment periodically? Or perhaps stay on some kind of maintenance therapy?
We just don't know if it's a one-time reset or if you need to keep topping it up to prevent slipping back to your old aging rate. That seems like the crucial next step to figure out.
It's the million-dollar question for the future of these reversal therapies. Persistence is key.